Jurre is a VP of Mass Spec Technology and Biomarkers at Olaris. He has 15 plus years of MS omics experience and draws upon that in his biomarker-driven research developments. One of his central aims is to translate omics-driven assessments into clinical biomarkers for improved precision diagnostics. His talk today sheds light on some recent developments at Olaris in clinical metabolomics using targeted MS methodologies. Jurre, the floor is yours.
Thank you. Let me just pull up my slides. Okay. Great. Thank you, Andrew, for the kind introduction and also the invitation to present some of our work here today. So my name is Jurre Kamphorst. I'm the VP of Mass Technology and Biomarkers at Olaris. And so I'll be talking about standardized metabolomic analysis of clinical samples using triple quadrupole mass spectrometry. So just a quick word about Olaris. So we combine metabolomics and machine learning to revolutionize how diseases are being diagnosed and treated. So we have a platform called Cerebro, which is a biomarker discovery engine that can be used to develop innovative precision diagnostics for earlier disease diagnosis, better selection of therapies, and response monitoring. So why do we like metabolomics so much? It's important to know that a number of assays that are currently being used in the clinical lab are actually based on metabolite measurements. So think about glucose for diabetes, creatinine for kidney disease, and cholesterol for cardiovascular disease. And I think part of why these tests are so useful is that they're so close to the phenotype of the patients. And so comparing it to other omics, genomics can tell you what could happen. Transcriptomics and proteomics will tell you what might make it happen. But metabolomics actually can tell you what is happening. And in addition to a person's genomic makeup, it will also capture the impact of the environment and the microbiome as well. So we're in the realm of diagnostics and clinical metabolomics, and for that work, we really like triple quadrupole mass spectrometry. And so there are like three key things to mention here. Why? So one is the data quality. So when it comes to sensitivity, linearity, and accuracy, triple quadrupole mass spectrometers are among the best, if not the best, types of instruments to use. Triple quadrupole mass specs are fundamentally targeted instruments, so you have to tell the instrument what metabolites to look at. And so that makes things easier at the back end. So it simplifies data processing. And so automated processing of hundreds of samples is becoming possible, which makes it conducive to precision medicine efforts where you're looking at large sample sets. Also, adaptation of standardized approaches across labs, including non-metabolomic specialist labs, also becomes possible. Then if you start to use these mass specs in combination with internal standards, you improve overall data quality. Again, if you want to compare large sample sets, including samples from different batches, you don't need those error-prone batch correction procedures. So I also say triple quadrupole mass spectrometry, even though it's been around for decades, still continues to develop and becomes quicker and quicker. So it's becoming possible to measure hundreds of metabolites with these types of instruments in a single run. So what goes into building a standardized metabolomics method? So there's three elements here: the selection of a metabolite panel, setting up your mass spec, and the use of internal standards. So for sake of time, I won't talk too much about the first two, but I will mention that for our panel, we've done a lot of work setting up the panel, and the current iteration for which we are showing the work here today contains 224 polar metabolites that we can analyze in a single run. In terms of LC-MS, we use helichromatography, a 20-minute method in combination with a thermal TSQ ALTIS triple quad. But I'll spend the majority or the entirety of the rest of the talk talking about 13C internal standards and what we were interested to see if it can actually improve our data quality by way of linearity of response, and also if we could use it to improve our normalization procedures post-acquisition as well. So 13C standards are really the kind of golden standard, internal standard for mass spectrometry. I will hasten to add that these are stable isotope labels, right? So these are not radioisotope labeled. That would be 14C. And so the use of these 13C standards allows to correct for sample-to-sample variability in sample preparation and analysis, as well as for matrix effects, which can be quite pronounced if you think about, or if you're talking about human clinical samples. And so 13C standards increase measurement accuracy and precision within, but also across batches. And so it improves also overall data quality. The fundamental issue I have with metabolomics is that you're measuring hundreds of metabolites. And so how can you possibly get all of the internal standards for all of these? And so getting these 13C standards individually is time not very time efficient and also cost prohibitive. And so the alternative here, and that's being offered by Cambridge Isotope Labs, is to use uniformly labeled 13C metabolite yeast extracts. And the idea here is that these yeast cells in a very controlled environment are grown on 13C labeled glucose, and they will produce uniformly labeled metabolites because of that. And so you use that yeast metabolite extract to then spike into each of your samples before the start of the sample prep. And so this is a very economic approach to address this issue. So we're very interested to test this and validate this. We've done a number of experiments, but I want to highlight one experiment in particular that we have done. So for our work, we are very interested in urine. Urine samples are great for diagnostics. They're obviously non-invasive. And also a lot of our work centers around kidney disease. And so the kidneys produce urine, and so that's the perfect kind of matrix or sample type for us to look at. From an analytical perspective, though, they're very challenging. They have very high salinity, and also there's quite considerable variation in hydration status of the individuals that donate these samples, and this causes large variations in sample concentration, and that is measured by specific gravity or SG. And so what we have done for this experiment is we took commercially available pooled urine, and we either diluted it 2 times and 5 times, but we also concentrated 2 times, 4 times, and 6 times. And so we have a range of samples that covers the entire clinical range of what we might expect in the clinical setting to see in terms of concentration of urine samples. So we used our the 13C yeast metabolite extract to spike into each of these samples at the start of our sample prep, and we looked at 2 things. One is the linearity of response across the sample concentrations, and then 2, see how we could use it to improve our normalization procedures post-acquisition. So this is looking at linearity. So what I'm showing here are 4 metabolites that are representative of different chemical classes. And what you see in this plot on the x-axis are the change in concentrations of the samples. And so we go from very dilute to very concentrated on the right. And so as you start to concentrate your sample, you expect to see an increase in the concentration of your metabolite as well, and it should be linear. But what you can tell from these plots is that for all of these metabolites, as these samples become more concentrated, the signal, if you look at the absolute MS intensity, becomes actually progressively non-linear, and that's an issue if you want to look for biomarkers. And especially in the case of arginine, we see some pretty interesting behavior, and that's not too uncommon for metabolites to see sometimes. But if we then use the respective 13C internal standards for these metabolites and express the data as a ratio to the internal standards, you can see that we see very linear behavior. I was actually surprised to see how well this works. And so what we can tell from this is that we get very good linearity across a 30-fold concentration or SG range with these 13C standards. So this is very nice data to have. So at this point, talking about numbers, we were able to detect in that panel of 224 metabolites for that panel, we were able to see on the order of 75 13C metabolites from the yeast extract. And so some of them are pretty low abundance. So we ended up having 50 what we call high confidence or high quality, I should say, 13C metabolites that we can use as internal standards in our study samples. And so we fully expect this number will increase as we further optimize our methods and further expand our panel going forward. But the question is, okay, for those metabolites that we don't have an internal standard for, what do we do with those? And so we looked into this idea of surrogate standards. And so the idea here is that for a metabolite that does not have its immediate 13C partner, we look for a metabolite that is similar to it, that does have a 13C internal standard. And so an example here is an acetylvaline, which has a retention time of 5.8 minutes, and it does not have its own 13C internal standard, but tryptophan is closely alluding to it, and we use 13C tryptophan as a surrogate standard. And you can see that with that we get good linear behavior using this approach. And so we wanted to look at this in a comprehensive manner across all our metabolites in our panel to see how well this actually works. And so we had to come up with a different kind of way of looking at this. R-square is great if you want to look at how well data points fit to your curve, in this case a linear line. But it's not great if you want to look at how the slope changes between data points. And so we came up with something else that's called change rate. The formula is shown here, and this is a mock example of what that entails. So let's say we have a metabolite and we look at it without a surrogate standard or with a surrogate standard. And so in the case of the orange, you can see that going from 0.5 to 1, you see a certain slope, and then going from 1 to 2, we see a different slope. And so there's a change in the rate. And so the change is quite considerable, and that is non-desirable. If we were to use a surrogate standard, then ideally what we would see is that going from 0.5 to 1, and then from 1 to 2, you don't see a change in that slope. And so the change rate will be 0. In other words, it will be linear, and that's desirable. So the closer to 0 you are, the better. And so we applied this to our entire panel, and the conclusion of that is that we were able to improve the data accuracy, linearity for an additional 105 metabolites using the surrogate standard approach, and that's in addition to the 50 metabolites for which we have an immediate 13C partner. And so the conclusion here is that the yeast uniformly labeled metabolite, 13C metabolite extract improves linearity of response for in total of 155 endogenous metabolites in our panel. And again, our panel is still in development, and this will continue to grow. So moving on from looking at linearity to normalization. So normalization, I like to think of as kind of a necessary evil. It's something we have to use for our samples to adjust for gross differences in metabolite abundances. In the case of urine, we need to correct for patient hydrated status for one thing. And so there's a number of ways you can normalize your data. I'm just highlighting one of them here, which is a pretty popular way of normalizing your data, which is median normalization. So you have your samples here, you perform LC-MS analysis, and then you end up with a data table with your samples here, and then your different metabolites at the various rows here. And so the idea then is that for each sample, you identify the median metabolite signal for that sample, and that becomes your normalization factor. And you express how you normalize this by essentially all your metabolites in that particular sample, you divide by that normalization factor. The key issue with this is that having one normalization factor per sample does not account for matrix effects on individual metabolites. And so different metabolites are affected differently. And so this actually generates quite a bit of artifact formation and noise. And so what we wanted to see is if we can do this, but with a little bit of a twist. So we still do median normalization, but instead of using metabolite intensities, we use ratios to their respective standards. So either their immediate 13C internal standard or a 13C surrogate standard. And that's what's shown here. But then we also take the median of that, but the median of the ratios. The benefit of that is that you're already accounting for some of the matrix effects because you're using these ratios to their standards. And so we wanted to see if that improves our data. So what we did is we actually used the very same experiments that I showed before, where we had the pooled urine and we started to make dilutions and also concentrated samples of that. And so what we're doing is, we've measured those and now we want to normalize those. And the idea is that because it's the same sample, you shouldn't see any differences post normalization. Of course, real life is a little bit different, so it's never perfect. But we wanted to compare the kind of standard media normalization versus our normalization using these ratios to the standards. And that's what's shown here. So these are individual metabolites. Each data point is an individual metabolites. And again, 0 is the ideal. You don't want to see any change. And what I hope you can see is that comparing the two is that with normalization using these ratios, you actually get improved normalization. So less of a spread and more density around the 0 line. So another way of looking at this is to apply stats post-normalization to the various concentrations of these samples. You shouldn't see any significant hits. If you do, they are false positives. And what you can see here is that with standard normalization, you do get some of those, and you only get one with our adjusted approach. So the conclusion here is that normalization using this ratio is definitely better than using standard normalization approaches and leads to fewer erroneous hits. So I'll just close with the questions like why do you care? Why do we care? Again, we're very interested in clinical metabolomics, and a key point to make here is that the bioanalytical emphasis really shifts from preclinical to clinical translation. So early on in the exploratory and preclinical metabolomics, those studies tend to rely on broad coverage and detection of unknowns. So this is the realm of high-resolution mass spectrometry and NMR. These tend to also be smaller, smaller sample sizes. However, as you start to kind of transition more and more towards the clinic, it becomes important to have high accuracy and reproducibility. And so this is where targeted MS comes in, and where NMR applications are also very useful. And so in essence, it's not just about quantity, it's also about the quality of the analysis as well. And so at Olaris, we use both NMR and LC-MS, and that combined platform provides accuracy and reproducibility at scale for hundreds of metabolites. So I'll close here and say thank you. I also want to thank the team at Olaris for all of this work, and I want to specifically highlight the Mass Spec team, Nihal, Yifang, and Masuma, and I'm happy to take any questions. Thank you.
Thank you, Jurre, for that excellent talk. We have time for a couple questions. So I had one, which leads into the question from one of the audience members. Oh, we have a couple more questions flooding in. To start, how was the 13C labeled yeast extract solubilized? How much was spiked into your samples? And then that leads into the question from Rong Meng as to whereabouts was it spiked?
Yeah, so we dissolved canonical with an extract into 2 ml of 50-50 methanol water, and there's 2 mLs. We used, in this case, 25 microliters of urine and 75 microliter of extraction solvent. So what we do is we, going from our 2 ml of yeast extract solution, we make a 5x dilution into the extraction solvent, and then we use 75 microliters of that to add to our 25 microliter of urine. And so what it ends up to is that we can use on the order, or we can spike in yeast extract into these samples for about 130 samples per conical of yeast extract. And so we really spike in this as early as possible. So as soon as we get these samples out of the freezer and they're thawed, that's when we spike in these. So before we do anything else to these samples.
Sounds good. All sensible, good practices. The next question is, How many types of metabolites do you use as internal standards to cover the entire detected metabolites?
How many types?
How many types of metabolites?
So in total, we have right now in the current version of the panel, we have 50 metabolites from the 13C yeast that we're using as internal standard, and they cover a pretty broad range of classes. So it includes organic acids, amino acids, some nucleotides, and sugars. So I would say it covers the entire range. Of course, there's differences in yeast metabolism and mammalian metabolism. So there are specific metabolites within each class that we may not see, or there may be substantial concentration differences. But it covers a pretty wide range of the chemical space, I would say. And I would also say that, kind of reinforcing this, that we have a panel and we selected certain metabolites for that. What we're going to expand this, and that will probably open up more, the use of more fertility metabolites from the yeast extract as well.
Fantastic. We look forward to hearing all about that in the future. So time is hit, so we'll need to move on to the next and final speaker of the session,